So today we'll be discussing about fluid mosaic model. So this question can come in your biochemistry exam or any biology exam. So the main answers about this point, the main points about this question you have to learn are what I'm telling you right now. The first one is you have to know what fluid mosaic model is. This model is briefly explains the cell membrane model. It was proposed by Singer and Nicholson in 1972. The membrane is made up of a lipid bilayer. What is a lipid bilayer? A lipid bilayer means there are two layers of lipids with proteins embedded. The proteins embedded can be used as transporters, enzymes, or receptors. Transporters, as you already know, there are different types of transports, like it can be through carrier protein, pumps, or ion channels. And receptors, it can be through hormones, growth factors, and neurotransmitters. The lipid membrane is amphipathic in nature. What does it mean to be amphipathic? Amphipathic means it has dual nature. It has a hydrophobic head, a hydrophobic tail, and a hydrophilic head. Structure is something like this. As you can see, this is the head part and this is the tail part. This head part is in contact with the aqueous environment and therefore it is hydrophilic in nature, and the tail part is in not contact with the aqueous environment, so it is hydrophobic in nature. So this forms a bilayer in the aqueous medium. The next is what kind of membrane lipids are there. There are three types of membrane lipids: phospholipids, cholesterol, and glycolipids. Phospholipids, what are the types of phospholipids? It is sphingomyelin and glycerophospholipids. And cholesterol, it is it provides fluidity to the membrane. And glycolipids, it is cerebrosides and gangliosides. Next we talk about the types of lipid movements. So there are two types of movements: lateral and flip-flop movements. Hence the membrane is fluid in nature. Why is the membrane is fluid in nature? Because it shows movement. What kind of movement? Lateral and flip-flop movement. Flip-flop movement is something like that. First we have this lipid and it flips like this. This flipping takes place, and lateral is like this. A ether and B is here, and it flips like this. A this side and B this side, flipping and lateral movement. And what are the factors which affect the membrane fluidity? Factors are cholesterol, which increases the fluidity, saturated fatty acids, which decreases the fluidity, and the amount of unsaturated fatty acid, if it increases, it also increases the fluidity. You know that the plasma membrane is mainly made up of phospholipid bilayer and is interspersed, means it carries or different types of proteins and carbohydrate residues attached to it, proteins and lipids. It can be glycoprotein or glycolipid. Cholesterol is also a component of animal cell membrane. And we have transmembrane proteins serves as receptors, tissue specific antigen, ion channels. Lipophilic molecule easily pass through the cell membrane, whereas hydrophilic molecules find it difficult to pass because hydrophilic molecule has polar nature and membrane is lipid in nature, and a lipid does not like polar molecules. It always repel the polar molecules. Like molecules attract each other in this case. So let's talk about the structures. As you can see, this is the integral protein. It is deeply embedded. And this is the channel protein through which transport of substances can take place. This is the peripheral protein, which is loosely embedded or loosely attached. This is glycolipid, which is carbohydrate plus lipid. This is normal carbohydrate chain. Glycoprotein means carbohydrate plus protein. This is globular protein and cholesterol, which provide fluidity to the membrane. Alpha helix protein, which also is a configuration of protein. So this model you can draw the diagram Internal surface, external surface, alpha helix is a similar diagram but in diagrammable form. So as we can see, there are membrane proteins which are of two types: peripheral and integral. Peripheral, which are loosely attached, and deeply embedded, which are integral. So it's a very broad classification. You may ask, what are the function of these proteins? I've already told you they act and they have many functions, but mainly they are of three functions: transport, enzymes, and receptors. Transport, they can act as ion channel, pump, carrier protein. Receptors, they can be hormone, growth factors, neurotransmitters, and next is the enzyme. Then we have the components of respiratory chains, as you know, ETS and all. One thing you need to understand that this fluidity helps the membrane to perform exocytosis and endocytosis. You must be knowing what endocytosis and exocytosis is. What is exocytosis? Can you tell? You have to recall that. What is exocytosis, endocytosis? It's a very important topic. Recall it and let me know in the comment section. The next is function of plasma membrane. What is the function of plasma membrane? So as you can see, the main functions of plasma membrane are: it protects the cytoplasm and organelles. If there is no cell membrane, then all the organelles and cytoplasm will get spread out through, and there is no boundary. There is a selective barrier, means it permits the transport of required substance in either direction. It helps in transmission of signal. It maintains the shape and size of the cell, and it helps in cell-to-cell interaction. Phospholipids in the cell membrane are sphingomyelin and glycerophospholipids, and glycolipids are gangliosides and cerebrosides. As you can see here, glycolipids are glycosides and cerebrosides. So this is what you have to write when a question comes of what is fluid mosaic model.